Hello everyone and welcome to the video. Today we are jumping into meiosis. So in our last video we did a little bit of review, talked about some related vocabulary to meiosis. We talked about some karyotypes. And now we're going to jump into meiosis, which is the reason why we talked about all these. So to start off with a general definition for meiosis. Meiosis is going to be the process that creates gametes for sexual reproduction. And as we hopefully remember, gametes are going to be cells that contain half the usual amount of chromosomes, and it has 23 single-stranded chromosomes in each cell. So in meiosis, our whole goal is to create these gamete cells. We're hopefully going to end up with gametes, and each one should have 23 single-stranded chromosomes in there. The reason we do this is because gametes are going to then combine during sexual reproduction to give an offspring a full set of genetic information. So as we remember, hopefully, gametes are going to be our egg and sperm cells. Each of them are going to contain half the usual amount of chromosomes. So a half plus a half is a whole. So our two gametes combine during sexual reproduction to give our new offspring a full set of genetic information. We're going to start looking at this picture over here. And I know there's a lot to look at, but this is not necessarily new territory for us because meiosis is going to be very similar to mitosis. Not only do these two processes sound very similar, but a lot of their kind of specific parts are also going to be very similar as well. So first off, meiosis does the phases of mitosis twice. So let's look at this picture to look at that first. If we remember, the four phases of mitosis are going to be prophase, metaphase, anaphase, and telophase. PMAT is our acronym for mitosis. So that's not new for us. However, what we are going to see, we do have a number that comes up here. So on our top part, we have prophase 1, metaphase 1, anaphase 1, and telophase 1. However, that's not the end of meiosis. We come down here and then we start the phases again. So we have prophase 2, metaphase 2, anaphase 2, and lastly, telophase 2. So the way that we differentiate between these is the number that we put after the name of the phase. So these numbers come really important when we're differentiating between the different stages of meiosis. So do pay particular attention to these numbers that come after these phase names. The next kind of similarity is that meiosis starts with double the amount of chromosomes compared to mitosis. So while there is similar looking structures here and similar looking processes, in prophase 1, it's maybe difficult to see in this drawing here, but in these homologous pairings here, we have 3 sets of homologous pairs. We have red and blue chromosomes. However, each of these are going to be double-stranded. It's easier to see here in anaphase 1 as these these double-stranded chromosomes are getting pulled to opposite sides of the cell. You can see that they're still double-stranded here. All these chromosomes here are double-stranded. So when we start off here in prophase 1, we're starting with double the amount than we normally would in mitosis. So if we take a look down here, we just have these double-stranded chromosomes. These two cells look much more similar to what we're used to dealing with in mitosis. So in the first set of meiosis stages, the first round of these stages of mitosis, we're going to have double the amount of DNA here. And then our last similarity, or rather a difference, is that one thing that happens in meiosis that does not happen in mitosis is this process called crossing over. So this process crossing over occurs during meiosis, and it's going to lead to genetic diversity. And we're going to break this down a little bit on the next slide and talk about how this process of crossing over allows us to create gametes at the end of meiosis that are going to be genetically different or genetically diverse from each other. All right, so now we're going to start to break down genes being on a chromosome. We've talked about genes before, we talked about chromosomes before, but now we're going to talk about how things get laid out and how things can actually get mixed up during meiosis. So the first thing to remind you of is that each of our 23 pairs of chromosomes contain different genes. So what this means, we remember 23 pairs of chromosomes in a human genome in all of our full genetic information. And each of our different chromosome pairs, for example, our first chromosome pair is going to have different genes from our second chromosome pair or a third chromosome pair. If we just had repeats on all of our chromosomes, we wouldn't really have a need to have 23 pairs of chromosomes. The reasons that we have 23 is that each pair of chromosomes contains different genes. It contains different genetic information. So that's the first thing to keep in mind. The information on chromosome 1 is not going to be the same information on chromosome 2 or chromosome 3. And when we think about these, for one chromosome, for example, the genes are going to be lined up in a certain order on the chromosomes. We're going to look at this first picture here. We can see we have these two single-stranded chromosomes, and we're going to think of these two as being a homologous pair. That means, if we remember from our last video, our homologous chromosomes are going to be two chromosomes that have information for the same genes on them. So for example, gene A is here, gene B, gene C, D, E, F. They're all going to be on the same chromosomes. So for example, we can think of gene A being hair color, gene B being eye color, and gene E being the height gene. So when we look at both of our chromosomes here, they're in a homologous pair together. So these two are in a homologous pair, and the genes are going to be in the same order. So it's A, B, C, D on the first chromosome and A, B, C, D on this other chromosome. So it's important to keep in mind that the order that genes fall in on a chromosome stays the same. However, gene and allele combinations on chromosomes in a homologous pair are randomized through crossing over. So the gene and allele combinations on a chromosome in a homologous pair are randomized through crossing over. What you may have noticed in this picture here is that although we do have the same kind of sections of our chromosomes set out for each gene, we do have some different colors here. So for example, for gene C, on this left chromosome, we have a red color, and on the right chromosome, we have an orange color. This is representing that even though both of these sets of information, these areas of the chromosome code for this gene C, there are different alleles here. We also have different alleles for gene A and different alleles for gene E. So what this is basically going to mean is that we can have information for the same genes here, but our two homologous chromosomes can have alleles. They can have different alleles, which means that these genes are going to have different precise genetic information there. And when we think about this, crossing over is actually going to kind of mix these alleles and chromosomes up. So the second part of this, chromosomes in homologous pairs exchange equal parts of their chromosomes with each other. And to point this out, I know that crossing over may be slightly specific. I'm going to link an extra video in the description of this video that will kind of help to maybe explain this more in depth. But to look at this, we're going to look at this example here. So we're going to have these two chromosomes. These two chromosomes are going to be in a homologous pair. But hopefully we'll notice is that our two chromosomes here, while their sister chromatids are identical, as they should be, the exact alleles on our different homologous chromosomes are different. So our pink chromosome here has big A alleles and little b alleles, while our blue chromosome has little a alleles and big B alleles. So our alleles on our two homologous chromosomes are different. What is more or less going to happen during the process of crossing over is the second picture here. We can see that on our blue chromosome here, there's now a small section of it that is the pink chromosome. They're quite literally going to cut off a small piece of each of these chromosomes, and they're going to switch spots. So this little piece of the pink chromosome is now on the blue chromosome, and that same little section from the blue chromosome is now on the pink chromosome. And eventually, once we get to the end of meiosis, these four chromosomes are going to get split up into our four single-stranded chromosomes that end up in our gametes. And what we can see here is that we have some new combinations of chromosomes. If we did not do crossing over, all that we would have are these two options here. We should have two that are big A, little b, and two that are little a, big B. However, because of crossing over, we have increased the genetic diversity. We have changed around the combinations. We now have one that is big A, little b, big A, big B, little a, little b, and little a, big B. So because of this process of crossing over, we have made new combinations of chromosomes. If we didn't do crossing over again, we would only have these combinations, big A, little b, and little a, big B. But because we did crossing over, we have increased our genetic diversity. We have created new combinations of chromosomes. And that's why this process of crossing over is so important. It helps us to increase our genetic diversity on our chromosomes. So genetic diversity, again, it increases, crossing over increases genetic diversity by creating different possible chromosomes in each gamete. Again, each of these single-stranded chromosomes, as we're going to talk about, is going to end up in a different gamete. So this is going to be in one gamete, this is going to be another, this is in another gamete, and this is in another gamete. And because we did this process of crossing over, we have created new possible combinations of unique chromosomes. And then crossing over happens differently each time it happens. And this last note here is really important. We're going to have different combinations and different parts getting switched out. So for example, in our first crossing over event here, we had this bottom part get switched out. But if we are creating new gametes, again, we could have the entire bottom leg of the chromosome. So this entire blue part can get switched out with this entire pink part. Or we could switch the top part of the chromosome instead. Crossing over is going to happen differently each time it happens, which is only going to further increase the possible combinations of chromosomes that we can make through the process of crossing over and through the process of meiosis. This is going to make a little bit more sense once we talk through the stages of meiosis, but keep this process of crossing over in mind. We are increasing genetic diversity by doing crossing over. All right, so as we mentioned on the first slide here, we're going to break meiosis up into two halves. So the first half that we're going to talk about is called Meiosis I. So this is the first set of phases. So the phases look very similar to mitosis, except there are pairs of double-stranded chromosomes. So as we'll notice here, you can see it pretty clearly in metaphase here. In metaphase and mitosis, we should just have a single file line of X's. However, we have these double-stranded chromosomes that are matching up in their homologous pairs so that they can do this process of crossing over. This is another example picture of crossing over for you. So we can notice here that our chromosomes did do some crossing over while they were in this metaphase area. And once they start to get pulled to opposite sides of the cell, we still have double-stranded chromosomes. Again, this is different from mitosis when we talked about the cell cycle, because in mitosis, we had our double-stranded chromosomes getting pulled apart into single-stranded chromosomes. In meiosis I, we still have our double-stranded chromosomes intact by the end of it. So the main thing you want to keep in mind for meiosis I is that this is where homologous chromosomes pair up and crossing over occurs. To give us a definition for crossing over, in case it wasn't completely flushed out on the last slide, crossing over is a process where homologous chromosomes exchange equal parts of their chromosome. So again, we can look at this example here. Our orange chromosome is paternal, it came from our dad, and our red chromosome is maternal, came from our mom. These two chromosomes, they're crossing over here. You can see they're crossed across each other, and they're going to exchange equal parts of the chromosome. So now this orange chromosome has a little piece of this red chromosome, and this red chromosome has a little piece of this orange chromosome. This is, again, this process of crossing over. When we're thinking about when crossing over occurs during prophase 1/metaphase 1. I know in this picture it really only shows that they're lined up in metaphase 1, but during prophase 1 they are going to start to move together and they're going to start to propagate and get into their homologous pairs. So this process of crossing over really kind of happens in this in-between stage as we're transitioning from prophase 1 to metaphase 1. So in this process, spindle fibers are going to pull our double-stranded chromosomes to opposite sides of the cell, creating two diploid daughter cells. So what we should notice here is that by the end, when we do cytokinesis the first time, we should have two cells that still have these double-stranded chromosomes in there. So again, I mentioned we create two diploid daughter cells. So again, we're going to have these double-stranded chromosomes at the end of meiosis I. We have two cells that both have double-stranded chromosomes. And if you do look at the detail of these pictures, we can notice that crossing over has occurred on these chromosomes. These chromosomes are not the same color that they were starting up here. So prophase 1, we just have blue and we just have pink. But down here, the blue and the pink got mixed together, representing that crossing over has occurred. Next, there's meiosis II. So this is where we repeat those stages, or rather those phases of mitosis that we've already talked about. as we should hopefully notice here, is that these pictures, it's the same process of mitosis from the cell cycle, except it's occurring in two cells at once. So if you were, for example, to just focus on this half of the picture, it looks exactly the same as mitosis. So we have mitosis. It's prophase, metaphase, anaphase, telophase, cytokinesis, just the same as we're used to looking at it. The difference, though, in Meiosis II is that we need to keep in mind this is happening in two cells at once. At the end of Meiosis I, we ended up with these two diploid daughter cells. But we need to make sure that these two daughter cells get properly split up. By the end of Meiosis II, the final product that we're working with is 4 haploid gametes that have been created. So by the end of Meiosis II, we have these 4 cells down here, and these are our gametes. They have half the normal amount. They only have 1 copy of each chromosome in them. And that was, again, the whole goal of us doing meiosis, is that we wanted to end up with these gametes at the very end. That was kind of our overall goal of meiosis. All right, so what I'm displaying here is just another set of pictures to try to kind of drive home what's happening in each of these phases of meiosis. So these are pictures of Meiosis I, so prophase I, metaphase I, anaphase I, and telophase I/cytokinesis. You may be able to see a little bit more easily here that we still do have double-stranded chromosomes. So if you look really closely, you can see some detail showing that even though it may not look like it, we still have double-stranded chromosomes throughout this entire Meiosis I process. So again, in prophase I, same as mitosis, our nucleus starts to disappear, our spindle fibers start to form, and we're pairing up in our homologous pairs for chromosomes. Metaphase I, we line up in the middle of the cell. Anaphase I, as in mitosis, our chromosomes are getting pulled to opposite sides of the cell. However, we do need to keep in mind in that anaphase I of meiosis, we do have these double-stranded chromosomes. And then in telophase I and cytokinesis, we still have double-stranded chromosomes. Our nucleus is kind of starting to reform. We have our cleavage furrow forming in the middle here, separating our 2 new cells. And then we're going to go right into Meiosis II. Again, if we remember, Meiosis II, this is happening in 2 cells at once. So you need to keep in mind that 2 different cells are doing these phases of Meiosis II at the same time. So prophase II looks almost the same as prophase from mitosis. It actually does look the same. We're going to have our nucleus disappear as before. Metaphase II, we have our double-stranded chromosomes lining up in the middle of the cell in a single file line. Anaphase II, our double-stranded chromosomes get ripped apart into single-stranded chromosomes, getting pulled to opposite sides of the cell. And then telophase II and cytokinesis looks very similar again to our final stages of mitosis. And this is going to be what we end up with. We're going to have 4 of these gametes, 4 gametes that hopefully should have these unique chromosomes in here. So what I'm going to ask you to do, if you haven't done so already, is I would actually suggest you can choose either set of pictures. You can use these 2 sets of pictures, or you can use these 2 sets of pictures. I want you to kind of pause the video at different points and try to copy these pictures into your notes. It's going to be good for you to practice drawing these, and be especially sure that in Meiosis I, we are making sure we are drawing these as double-stranded chromosomes so that we remember throughout all of Meiosis I, we have double-stranded chromosomes. So go ahead, choose whatever set of pictures looks better to you, and draw these in your notes just so you have them for reference. Alrighty, thank you all for watching.